Hello, good afternoon, good morning, or good evening, wherever you are in the world. We welcome you to the second part of the ISTA Plant Health Seminar. The first part was last week, Wednesday, taking care about regulatory aspects on plant health. And today we go into the second part with the new threats, outbreaks, and regulations. And we welcome our 5 speakers here on the screen. Please note that this broadcast will be recorded and it will be made public also on the ISTA YouTube channel afterwards. If we have time for questions in the end, if it is not very much after 6 o'clock Swiss time, we will also allow questions, which I would kindly ask all attendees to put in the question and answer box, and we will try to answer all these questions. Please do me the favor, and if you have questions to a person, please mention to which presentation it is going to be addressed. My name is Andreas Weiss. I'm Secretary General of the International Seed Testing Association, and I also welcome Valerie Grimaud as the chair of the ISTA Seed Health Technical Committee. Now I will introduce the first speaker, which is Robert Gilbertson. He is a professor at the University of Davis, the College of Agriculture and Environmental Science, and his talk has got a very short topic. It is TOBRFV. And Robert, you will tell us probably what these 4, 5, 6 letters stand for.
I will. Good morning, everyone. Let's see. Let me see if I can share my screen here.
And I would like all other panelists, please go on listen mode and close down your cameras.
Okay. Let's see. Can people see my screen? Can people see my screen?
Not yet, Robert.
Not yet.
Okay. Let's see. I clicked show my screen, and let's see. I don't know why it is not.
Go on sharing on that and on show.
Sharing, okay. Okay. Sorry, I don't see show. I apologize. So if I go to sharing, okay, show screen.
There we go.
Okay. All right. Thank you. I have not used this yet. Okay. So, all right. All right. Okay, everyone. Well, good morning. And again, my name is Bob Gilbertson, and I'm a seed pathologist virologist from the University of California. And I'm going to talk to you today about tomato brown rugose fruit virus, but I'm going to try to talk about how the knowledge of this group of viruses, which are called tobamoviruses, can help us understand and manage this new emergent virus, tomato brown rugose. All right, so first, those of you who are wondering what is a tobamovirus, let me give you a very brief background on this. So tomato brown rugose fruit virus is a member of the genus tobamovirus, and this genus, like many genera of organisms, is contained in a family called Viridae. And the tobamovirus type species, all genera like tobamovirus have a type species, is tobacco mosaic virus. And anyone who knows plant pathology knows tobacco mosaic virus because it's a very famous plant virus, probably the most famous plant virus. In this family, however, there's 37 currently recognized species, including brown rugose, and currently recognized means there's probably more out there. So we discover more of them all the time. Now, all members of this genus of virus, tabamovirus, are going to share similar features, and the most diagnostic feature is shown at the bottom here, this distinctive rigid rod-shaped virus particle. So if you purify the virus from the plants and view it with an electron microscope, that's what you'd observe. If you stripped away the outer shell of protein, you'd have one single RNA molecule plus sense about 6.4 kb. Okay, so this is a plus sense single-stranded RNA virus like most plant viruses are. And of course, if you're everybody knows the most famous plus sense RNA genome virus out there is the coronavirus, which is, of course, much larger, greater than 30 kb. Okay, so all of the members of these viruses, tabamovirus, have similar genetic properties. Their sequences are similar, their gene organization is similar, and so on. And then they share biological properties, and most notably, these viruses are spread by contact, by touch, by touching plants, by any sorts of contact, and by seed, as I mentioned, but there's no insect vector, okay? Now, you'll hear bumblebees can move it around, and that's true, but that's just contact. They're not true vectors, okay? And of course, these viruses cause a number of economically important diseases. Now, many of them, or a number of them, infect tomatoes, and those are the ones we're going to talk about today. And in particular, we're going to talk about tomato brown rugose fruit virus, because this is a new emerging virus that has become a big, serious problem in protected agriculture, as I mentioned. So if we look at the ones that infect tomato, actually 2 of them are emergent, tomato mottle mosaic, which I'll briefly mention, but tomato brown rugose fruit virus is the main problem these days. It's a newly emergent virus, first appeared in 2015, and it causes very similar symptoms in the foliage. It was notable because it induced these necrotic lesions in fruit, but only certain varieties show this necrotic fruit lesion from which the name comes. And nowadays, growers have figured out what those are and don't grow them. So you don't really see this brown rugose symptom that much anymore. But the reason it's become particularly important is because it has broken this important dominant resistance gene, TM22, that had been introgressed from a wild species into tomato and had been widely grown in protected culture to protect against these types of viruses. But unfortunately, like many pathogens do, this pathogen has evolved and is able to break this resistance gene, and this has created a lot of problems in the industry because of the dependence on this TM22 gene. All right, quickly, I want to mention, when we talk about where rugose is, tomato brown rugose is a problem, it's very easy to focus on protected culture, okay? And I'll talk about why this is. Where we see this problem is in these high-tech greenhouses, either in temperate areas like Europe, for example, okay? Or in high-tech greenhouses or greenhouses in tropical or more tropical areas, for example, Mexico, where this is a big problem, okay? So these are really the areas that we see this. Now, I work a lot with the processing tomato industry in California. And if you know this industry, you know the big virus problems there are not tomato brown rugose, but the resistance breaking tomato spotted wilt and beet curly top virus. So in our outdoor processing tomato production, we don't touch the plants hardly at all. We have in California a 3-month tomato-free period during the winter. So this virus has even yet to be found in our outdoor processing tomato. And as I tell our growers, I don't believe it's ever really going to be a problem. In fresh market production, it can also be a problem in outdoor circumstances and in certain high-tech, in lower-tech greenhouses as well. And I'll mention that at the end. All right, so how do these viruses move in a plant once they're introduced? Okay, now this little cartoon shows you a virus being introduced by an insect vector, of which 80% gain access in that way. But as I mentioned, tabanoviruses get in through tiny little wounds. And once they get in, they have to do the same thing that any plant virus does. They first have to get in, they replicate their genetic material, then they have to move out of the initially inoculated cell. Now remember, all plant cells have this thick cell wall, and you can't move through it readily, only very small molecules. So plants have evolved these little pores that interconnect cells that are called plasmidesmata. And the viruses, of course, have evolved to move through these pores, which are called plasmidesmata, and they move from cell to cell in this way. We call that cell-to-cell movement. Once they access the vascular system, and here you can see this cell-to-cell movement going on into the vascular system here, then they move long distance rapidly through the plant, okay? And this is where the trouble begins, okay? Because they can spread throughout the entire plant and start inducing disease symptoms because they take over the plant and they trick the plant into making virus and more virus nucleic acids and proteins. And then eventually you see the plant entirely infected. And if you're lucky, sometimes you catch the infection right in the middle of a leaf going from being a source of photoassimilate to a sink. And you can see how the virus can still go in where photoassimilate is delivered, but no longer can access the exporting tissues that are exporting photoassimilate. So it's a very interesting system how the viruses move within plants. Once in the phloem, they move very quickly, and what they systemically infect the plant throughout the entire plant, and then some sort of symptoms are induced, like this mottling and leaf crumpling that you can see induced by tomato brown rugose fruit virus in tomatoes, okay? And then once in the phloem, they move to what we call sink tissues. And these are tissues that are demanding photoassimilate from the plant to allow them to grow. And the two main sinks are the roots, and remember the roots, okay? Because people don't think about it. There's a lot of virus that goes down to the roots in infected plants. And then the shoot apex is the newly growing kind of a young tissue of the plants that give rise to the leaves, okay? And so that's what the plant is delivering food to, and the virus is just going with the flow, but that's where it wants to go anyway. And then it gets into cells and goes through the life cycle, as I mentioned. And then at some point, there needs to be plant-to-plant spread. And in this case, somebody touches an infected brown rugose plant and then touches a healthy plant. That's all you need to do to spread it. So this virus, by contact, can spread like wildfire, okay? So again, the virus moves in the direction of the sinks. It's very passive. It goes with the flow, like I mentioned. And in about 7 to 14 days after delivery up into that shoot apex, this is what you see, okay, these symptoms. Now, of course, you don't see symptoms really in the roots, but I can assure you they're down there. And by the end of the infection, the virus forms these virion particles, these rigid rods that I mentioned before, and they pack together in these crystalline structures called inclusion bodies. And hundreds and hundreds of cells are packed with these inclusion bodies, which are packed with thousands of these virus particles. So imagine how much virus is in one single plant. Now, some really classic work was done on tobacco mosaic to follow the movement of it in tomato plants. And you can see this was done by Samuel in 1934. And they used indicator tobacco plants to indicate where the virus is, because the tobacco plants make local lesions, which I'll show you in a minute. And what they found was it took the virus a while, a few days, to build up in the inoculated leaves, about 3 to 4 days. And then, as I mentioned, it moved right down to the roots by 4 days. By 5 days, it was up into that shoot apex and extensively colonizing the shoot apex. Continued to do that through 12 days, symptoms show up, and by 25 days, the entire plant, this is a young plant, was colonized by the virus. So you're talking about a virus that moves fairly quickly and eventually can be found throughout the entire plant. And again, as I mentioned, there's huge amounts of virus in there, okay? So, this is a big challenge when it comes to this virus getting introduced because it builds up quickly and spreads so fast. All right, so let's talk about the biology of tabamoviruses and then how do we try to manage it, okay? Now, as I mentioned, tabamoviruses are known to be seed transmitted, and now we're talking about ones of tomato, okay? And it's also known that they are contaminants on the outside of the seed. All right, now they move up into the sinks, as I mentioned, and the fruits become a sink. But by the time the virus gets into the fruit, the seeds have already been established and their vascular connections have broken down. So this virus never enters or infects the embryo, all right? So it's not truly seed infecting, all right? But it's a seed contaminant, all right? And some people will argue, well, it's pretty inefficient at getting from the outside of the seed because, you know, the seed has to germinate and the little hypocotyl has to rub along the outside and there has to be a virus particle. But as I'll show you in the next slide, tomato brown rugose has found a very nice way to be able to get from seeds to plants. We don't know if it's changed its biology. I doubt it has. But again, it's not as inefficient as people, seem to say, okay? And as I'll mention, the worldwide spread of tomato brown rugose is evidence of that, all right? So this tells us that seed becomes a critical component for these viruses, and we know this from other ones, okay? And the RT-qPCR test that has been developed to detect this virus in seed is an excellent test, and now is being commonly required for transplant greenhouses to produce a seed lot. For example, in California, our transplant growers are requiring this test has been run on seed lots or they won't use them. Now, as I'll mention later, quickly, you can use a tobacco bioindicator to really help this. You can also disinfest seeds with various things. In our lab, we use 10% triple sodium phosphate for 30 minutes, but there are other ways. Okay, so there's ways to not only detect it in seeds, but eradicate it as well. And, you know, now people are learning, don't take seed from people you don't know, okay, or from areas that are far away, because this is the way it also can get it. And here's just where we found the virus most up to date, most recently, and these red dots. And it started out in Jordan and the Middle East, and you can see how it marched right along, and all of you realize it's following right along with the seed kind of production pathway or pattern for protected culture, okay? So this is totally consistent with that. Now, as I mentioned, we have excellent tests for this virus now, this RT-qPCR, so we're in very good shape now for being able to detect it. Like I say, I like this touch of inoculating this biological indicator, but obviously you have to have a greenhouse and you have to grow plants, and the industry's moving away from this. But as a plant pathologist, I like that biological touch. Now, transplants. Most tomatoes today are grown as transplants, and in California, they're grown in these giant transplant houses, okay? And as I just told you, it takes a while for the virus to get from the seed to the seedling, and then it takes time, 14 days to get symptoms. So you're not going to see much symptoms in there. And good luck walking through that transplant house and trying to monitor for it, okay? Because it's kind of difficult. So there again, the tested seed is critical because it's very hard to walk through this whole house. But can transplants be a source? Absolutely. And in 2020, transplants were brought in from Mexico. How they got in, I don't know, to Arizona, and they had a huge outbreak of tomato brown rugose in these Arizona protected culture houses because they brought transplants in from Mexico, which if they had asked anybody, they would have said that's a very bad idea, okay? So again, use the certified tested seed. There's a good test out there, okay? Then, of course, you can, it's very difficult to test the transplants, but you can follow them out into the houses. And if the plants show symptoms early, then you're suspecting, maybe they came in with the transplant. All right, now, when you're in the protected culture, and I'm really going to talk about protected culture here because we don't regularly monitor our processing tomatoes like this, but in protected culture, you must do this because you need to know when the virus comes in. And so you need to be out looking for the symptoms in those newly emerged leaves, like I just told you. And today, there's good lateral flow tests for tobaccoviruses, and even the brown rugose one is apparently available now, okay? So you can confirm if you see symptoms with the rapid lateral flow test, okay? And so this allows you to get a quick idea when it's introduced, and then following the pattern, you get some insight into how it got in. But let me emphasize, whoever's on the team who's walking around and looking at the plants needs to know what they're looking for, okay? Now, soil. I told you that a lot of this virus goes down to the soil, and this virus is very stable. These rods are extremely stable. So the virus will persist in soil for years, as long as there's debris. So you either have to rotate, you can't really treat the soil, you might be able to use resistant rootstocks. As I'll mention, resistance has been identified, but many houses are using the soilless culture and not messing with it. Now, in outdoor production, the virus can be maintained in reservoir hosts, but again, it's not been a big problem. Here's the type of patterns you look for. If it's all over, you know you're totally basically screwed. But here's kind of random, which if you see it in this way, it probably came in with the seed. If you see these clumped distribution, it probably persisted with soil. All right. So again, one of the big goals here is, testing and knowing where the virus is found, and commercial fruit becomes a big issue. Commercial fruit gets loaded with the virus, no question about it. But as I told you, the necrotic symptoms are no longer observed. So what you see is basically kind of blotchy, irregular, nondescript symptoms, or with the new resistant varieties, you don't see symptoms at all, okay? So the issue is, are these fruits moving around with this virus a threat for the virus to get out and get into commercial production? Well, the virus doesn't ooze out of the fruits, okay? But if you damage them, which is pretty easy, you release the virus, okay? But there's been a number of studies done in Europe where they have tried to follow contaminated fruit by local consumers to see if there's any evidence that it would get into their gardens or things, and have found no evidence of that. So what is the probability of this happening? Personally, I think it's very low. But as many of you know, we have this testing that's going on at the border of the U.S. trying to stop this virus from coming into the U.S. from Mexico and Canada. And most of us think now with these resistant varieties that don't show any symptoms, that this is probably unlikely to be very effective if it ever was. Okay, so, all right. So, for example, we're finding it coming into California. Chet Karowski, a buyer, went to a local store in Sacramento, saw this marbling fruit, which could easily be irregular ripening from whitefly feeding, tested it with the tabamovirus strip, PCR'd it up, 99% identical to rugose. So it's coming in, not only to California, but other states as well. All right, I want to wrap up very quickly with disease resistance. People have found resistance to this virus, okay? And there's two types. All right, I won't go through everything here, but I want to remind you all, when you say tolerant variety in plant pathologist terms means it gets infected and shows symptoms, but still yields, okay? Now, if there's a variety that gets infected but doesn't show symptoms, that's not tolerant. That's actually resistant, but we call that intermediate or moderate resistant, and that has been identified for brown rugose, but so has strong resistance, where often single dominant genes prevent the movement of the virus, like the TM22 gene, okay? So tabamovirus resistance is a very important management strategy. Of course, the single dominant genes, from wild species are still probably the place to go. The TM22 is really durable, but we need to find additional ones, okay? So again, growers learned early on to grow determinate varieties, cherries, when this virus came in. Now, as I mentioned, there are these indeterminate varieties that are symptomless, so they have intermediate resistance. They've been commercialized by a number of companies, so they're available now. They're being grown in Israel and Mexico, and the losses now have gone from 100% to maybe 20% in both Mexico and Israel through the use of these varieties. Now, we have found through our limited inoculations with rugose in diagnostics that there are some resistance genes that confer very high resistance to brown rugose, and I know companies are working on that right now. So, there's a lot of good news coming out in terms of brown rugose, including many good diagnostic methods like regular RT-PCR. We've developed a LAMP test that can detect rugose in as little as 1 hour, okay? And this was an outbreak in Southern California that was very interesting. Here's your general RT-PCR for tabamoviruses. Here's a number of samples from this field in Southern California. And here's our rugose-specific primers. Well, 3 of them obviously were infected with rugose. What about these other ones? Turned out to be model mosaic. So we have a mixed infection down there. All right, so let me wrap up. Where are we going with brown rugose? We've got great detection tools and good disinfestant methods. Kai Ling has done a great job identifying those, okay? Emphasis on the seed is critical, okay? And will the RT-qPCR test suffice? I think it's looking very good, okay? These moderate resistant varieties have allowed the industry to bounce back where it was hurt, these protected culture situations. And you can prioritize very, I think, easily where you want to put your emphasis on detection. Obviously, it's in the seeds and during the production, very important. And then at the end, sanitation and so on, okay? So, all right, that's a lot easier said than done to implement all that. And again, I hope that the industry is moving in the direction of standardizing testing and how we look at this virus, because honestly, I think we're learning how to manage it, and I hope it's going to go the way of Pepino mosaic, where now, you know, we don't think that's going to wipe out our industry. All right, well, thank you very much for the opportunity to give this talk today. I'd like to thank my postdoc, Tomas Melgarejo, in my laboratory, who has done most of the work. Kati Cespedes has helped also. So thanks again, and I'll wait for any questions at the end.
Thank you very much, Robert. That was very interesting. We move on with the presentations. The next in line is Rob Tanner. Rob is from the European and Mediterranean Plant Protection Organization. And Rob is working in Paris as an Englishman. He will be presenting on assessing the risk of invasive alien plants on plant health for the EPPO region. And Rob, the stage is yours. If you want to switch on your camera, that would be great. And Robert and I will disappear with our cameras.
Thank you. And you can see my PowerPoint presentation?
Perfectly, yes.
That's brilliant. Thank you very much. So thank you very much for giving me the opportunity to present at this webinar. It's a very interesting array of presentations. And I'll be introducing to you today the work that EPO conducts on invasive alien plants and assessing those species for the EPO region. So the European and Mediterranean Plant Protection Organization was originally created in 1951. We now have 52 member countries, and our role is to support national plant protection organizations of member countries. We support these MPPOs in cooperation and harmonization of work, with producing regional standards on phytosanitary regulations and plant protection products, and information dissemination and networking through technical meetings, through international conferences, through sharing information and the dissemination of this information. As you can see from the bottom of this screen, we disseminate this information in a number of different formats through the EPO website, to the EPO Global Database, to the EPO Reporting Service, and the EPO Bulletin, producing scientific documents from authors and publishing of our standards. Our main remit is plant quarantine, plant certification and regulation of non-quarantine pests, invasive alien plants, biological control agents, and efficacy of plant protection products. Today, I will be focusing on our work on invasive alien plants. Invasive alien plants are numerous throughout the EPO region, globally as well. And basically, there are hundreds of ornamental species imported into the EPO region each year. And many of these species sit very quietly, very nicely within botanical gardens or private gardens and behave exactly as you want them to, and are just showy for their floristic prettiness. But some, unfortunately, jump over the garden fence and can become invasive within the natural environment, and also have negative consequences on agriculture and biodiversity. Hence the formation of the EPO panel on invasive alien plants in 2002 to evaluate issues with invasive alien plants, to collect information, to risk assess, and to prioritize different species.
Rob, just a quick thing. We see your screen with next slide and notes. Could you make the presentation a little bit larger for us?
I'm sorry about that. So you don't see the full screen?
We don't see the full screen.
Let's see if I can change that. Can you see the full screen now?
It is still that we see also the next slide and the notes. If it doesn't work, then no, it was even better before.
Was it? Sorry about this. I have two screens, and is that better?
Not really. So let's try to go ahead with it then.
Excuse me, Rob, I think it's the other screen which should be shared.
Is that better?
No.
Is that better?
No.
Oh, I'm so sorry.
Rob, do you want me to share screen?
Yeah, go ahead, Olga.
Yep. Okay, so yeah, as I was mentioning, so there are a number of impacts that can be associated with some of these plant species that do have invasive tendencies and do enter the natural environment or the urban environment, including impacts on biodiversity, impacts on agricultural systems, invading of pasture land, invading of orchards, or reduction of yield for vegetable crops, etc. There's also impacts on ecosystem services, including provisioning ecosystem services, for example, the reduction of genetic diversity within the population, the impact on regulating ecosystem services, for example, soil formation, pollination, water regulation or air quality, cultural ecosystem services. Plant species can invade water habitats or colonize riparian systems, reducing the aesthetical value and cultural heritage of habitats, reducing access to water, to land, reducing tourism, and supporting ecosystem services, changing nutrient cycling in environments, etc. Next slide, please. There are a number of pathways that these species can enter, and I've mentioned the horticultural industry as one pathway, but there are also a number of other pathways that invasive plants and their propagules can enter the risk assessment area, the ecoregion, for example. These can include grain, contaminant of grain for animal feed, contaminant of seed mixtures, contaminant of agricultural machinery and equipment, hay, manure. Invasive plant seeds can become attached to clothes and camping equipment, for example. Livestock wool products, and of course natural spread as well. Next slide, please. So to assess and evaluate these species, there are a number of regulations within the EPO region. It's important to note that EPO only makes recommendations, and it is up to member countries nationally to define these plant health regulations. And of course, we have the two common markets, the EU and the Eurasian Economic Union as well. We have the plant health regulation, but also we have in Europe the invasive species regulation as well, which regulates invasive alien plants based on their impact on ecosystem services and biodiversity. Next slide, please. Under the international plant health context, we have the sanitary and phytosanitary agreements, and this is the sovereign rights of countries to establish phytosanitary measures to protect plant life or health, but the measures should be technically justified. Next slide, please. And to assess the risk of the invasive alien plants, we would conduct pest risk analysis on these invasive alien plants. The ISPM 5 IPPC definition of pest risk analysis is the process of evaluating biological or other scientific and economic evidence to determine whether an organism is a pest, whether it should be regulated, and the strength of any phytosanitary measures to be taken against it. Next slide, please. EPO conducts this pest risk assessment using the EPO standard PM55, the express scheme standard. And basically, the pest risk analysis has three main components: pest risk assessment, initiation, the total pest risk assessment, and the pest risk management section. Under the pest risk assessment, you will be discussing reasons why you are performing the pest risk analysis, and you will be defining the pest risk analysis area. Under the second stage, you would be evaluating the taxonomy, the pest overview, host plants or habitats where the species can evade and become a problem. Is there any need for a vector? The current global distribution of the species, and as we've already mentioned, the possible pathways for entry for that species. Also, you'll be evaluating the likelihood of establishment, both in the natural environment and in protected conditions in the PRA area. You will be analyzing the potential for spread within the natural environment, and in the PRA area, both by natural spread and by human-assisted spread. You'd be looking at the impacts of the species. First of all, in the current area of distribution, because often this information can be taken and used for consideration within the risk assessment area, especially where a species is not currently present in the risk assessment area, but also looking at the potential impact of the species in the pest risk and assessment area. And you would be identifying the endangered area, where in the PRA area is the species most likely to have the most economic impacts. Next slide, please. All of the information collected is evidence-based. This can be through scientific information, scientific literature, grey material, reports, also expert opinions as well. What we really need to establish within the pest risk analysis is what we know. It's useful to know what we don't know, and what is also likely to happen to occur. Predictive models are very useful to look at the spread and the establishment of an invasive alien plant species. Next slide, please. As I mentioned, predictive models, often we conduct modeling, species distribution modeling for the species to establish, for example, in the top left-hand corner, where the species will become potentially invasive, and this can help us identify the endangered area. Next slide, please. The third stage of the pest risk analysis is phytosanitary measures, pest risk management. And this is to define what measures can be taken against the pathways or existing populations within the pest risk assessment area to mitigate the impact and mitigate the risk of the species. Next slide, please. So that's a very whirlwind example of how we would conduct a pest risk analysis for an invasive alien plant, but we're still stuck with a problem that we have a lot of plant species to potentially evaluate, and therefore somehow we need to prioritize the most problematic species to conduct a pest risk analysis against. A pest risk analysis is a time limit, an expensive process in the fact that it takes time and it takes expertise. We conduct pest risk analysis in EPO by inviting experts on the species, and we sit in a meeting room for 2 or 3 days in the form of an expert working group and thrash out, amend all of the documentation, conduct scoring on different sections. So it's a time-consuming process, and time-consuming also equals financial as well. So originally, we would identify a plant species that is identified as a risk either through the panel on invasive alien plants or through the EPO Secretariat, and we would include this plant species on the EPO alert list. We would also include an article in the EPO reporting service on the species, and the inclusion of the species on the EPO alert list is to identify this species to member countries, highlight it as a risk, but also for want for additional information. Highlighting the species to our member countries might also act to have additional information sent to EPO on the occurrence, on potential impact, etc. Next slide, please. But EPO also prioritizes the potential for invasiveness and for risk assessment through the EPO prioritization process. This is a standard PM56 and it's available on our website for use. And the EPO prioritization process is designed to firstly produce a reference list of invasive alien plants that can establish or that are established or could potentially establish in the EPO region. And the second component of the prioritization process is to determine which invasive alien plants have the highest priority for pest risk analysis. The prioritization process is in 2 parts, and I'll walk you through these 2 parts now. Next slide, please. The first part is to determine the potential invasiveness of the invasive alien species, invasive alien plant. We have 3 categories here: high, a list of invasive alien plants; medium, observation list; and low, minor concern. As you can see the matrix there, the matrix is dictated by spread capacity and impact. So the high spread capacity with a high impact would dictate that the species is added to the list of invasive alien plants. Only those species that are added to the list of invasive alien plants can then be taken forward to the next stage, where you would evaluate the priority for pest risk analysis. Other species with a lower spread capacity, a lower impact, would be included either on the observation list or the list of minor concern. These lists are not static. Species can be moved between the lists when additional information is published or comes to light, and these lists are reviewed periodically. Next slide, please. The prioritization process would then consider if the species has the potential for pest risk analysis by looking to see if there are additional if the species is traded internationally, if there are other pathways that can be considered, if there's pest risk management that can be implemented against these pathways, is the risk of introduction by these intentional international pathways higher than natural spread? And is there a significant area still available for the species to establish in the ecoregion? If all of these answers are yes, then the species could have a priority for pest risk analysis. Am I running out of time now? Just a quick example of two species that have been recently risk assessed within the ecoregion. Both species native to North America have agricultural impacts on yield loss. The pathways were evaluated as contaminant of seed, contaminant of grain, and a contaminant of seed mixture. Next slide, please. As you can see, there's the global distribution of both of the species, both of the Amaranthus species. They mainly have transient populations within the ecoregion, but there are established populations as well, and they are herbicide resistant, which also adds to the problem of the species. Next slide, please. The two species were risk assessed with a high phytosanitary risk to the ecoregion with a low uncertainty, and the conclusion was to add these species to the EPO A2 list, species that are present within the ecoregion but within a limited geographical distribution. And I think I'm coming to my final slides. And talking about the phytosanitary measures, a number of phytosanitary measures were suggested, recommended by the expert working group and endorsed by in the EPO process. This includes potentially for grain should be produced in a pest-free area, or grain has been sampled, including to according to ISPS 31 and inspected, or grain has been devitalized according to appropriate method, and similar measures were suggested for seed and for seed mixtures and native seeds. I think that is, oh yeah, and this is just a list of invasive alien plants that are recommended for regulation within the ecoregion, A1 species being absent from the ecoregion in the natural environment, A2 species, as I've already explained, present but with a limited distribution. And I thank you all very much for your attention and acknowledge the work of all of our experts within the ecoregion. Thank you.
Thank you very much, Rob. Yeah, when I pop up, it means that the time is coming to an end. Thanks a lot for your presentation. It was very useful. I've got one question here, which we talk about in a minute. And I now like to invite our colleague Marianne Loiseau from ANSES to present us about Candidatus Liberibacter solanacearum epidemiology, current state of knowledge. The last part of that presentation title was easiest for me, but Marianne, you will guide us through your whole presentation. Thank you very much.
Okay, thank you. Thank you. First of all, thank you to ISTA to invite me to give this talk about Candidatus Liberibacter solanacearum epidemiology. I will call it LSO for more easy talk after all. My presentation will be divided in 4 main parts. First, taxonomically consideration, then the geographical distribution of LSO. In the third part, I will present its host range and symptoms induced. Finally, I will present the current knowledge concerning transmission pathways of the bacterium. LSO was first described from Solanacearum plants and psyllids almost simultaneously in 2008 in New Zealand and the USA. The name Candidatus Liberibacter pylorus was initially proposed by Hansen, but Candidatus Liberibacter solanacearum, proposed by Lifting, was finally retained. As you can see on this phylogenetic tree, LSO is closely related to the Liberibacters associated with Wang-Lang-Bing, the most destructive disease of citrus. The Liberibacters belong to the alpha Proteobacteria class. The Liberibacters are phloem-limited and gram-negative bacterium. Until now, LSO has not been cultivated in arsenic medium. Thus, they are restricted to their host phloem and their vector's hemolength. The bacterium is genetically diverse and 12 haplotypes have been described. These haplotypes differ in their host range, psyllid vectors, and geographical distribution. In particular, 4 haplotypes, A, B, F, and G, are associated with disease of potatoes and other solanaceous plants, whereas other 4 others, C, D, E, and H European, are associated with disease of carrots and other apiaceous crops. Further described haplotypes, H North America and U, were not associated with a disease and were described in plants of the families Convolvulaceae and Urticaceae respectively. Haplotype H North American, which does not appear on the presented tree, was reported in Mexico and is different from haplotype H European, reported in Northern Europe. LSO cras, that you can see here, was isolated from psyllid Craspedolepta. In America, here, LSO A and B have been found from Nicaragua through Central America, USA, and Canada. More recently, the bacteria was detected in South America, in Ecuador and Colombia. LSO-R was also introduced in New Zealand by infected psyllids. LSO-C was introduced in New Zealand by LSO-C has been found in Northern America. LSO-D and E occur in Southern Europe and the Mediterranean region. However, the distribution of LSO could be wider than that which has been reported. Indeed, while testing collection of apiaceous seeds, Monger and Jeffries detected the bacterium in seeds coming from countries that had not reported it. You can see them here: Czech Republic, Denmark, Egypt, Japan, Lebanon, the Netherlands, and Syria. And we also have reports in carrot seeds in USA, in New Mexico state. LSO is known to predominantly infect solanaceous species, mainly potato and tomato, but also other economically important crops, and several wilts in this family. Here you can see few symptoms of zebra chip in potato. Sorry, I have problem with my screen. Okay. So leaves on younger shoots display purpling. Here you can see them, and the older one are chlorotic. You can also have aerial tuber formation, and the tuber symptoms include browning and vascular tissue. Upon trying, these symptoms become more pronounced, and crisp and chips processed from affected tubers show very dark blotchy strips and streaks, rendering them commercially unacceptable. The symptoms in potato tubers has led to the disease being named zebra sheep. In Europe, this bacterium affects Apiaceae species including carrot, celery, parsnip, parsley, fennel, chervil, and several wilts in the family Apiaceae. Symptoms in carrot infected with LSO include leaf curling, yellowish, bronze, and pearlish discoloration of leaves, stunting of the carrot shoots and roots, and proliferation of secondary roots. Similar symptoms of vegetative disorder are observed on other Apiaceae crops, but the infected plant can also be asymptomatic. Different reports suggest that the host range of LSO is larger than the Solanaceae and Apiaceae families. Indeed, in a laboratory study, 2 plants belonging to the Convolvulaceae family, sweet potato and field beanweed, were tested positive after transmission trial with Bacteria coquerelli. LSO was reported in stinging nettle and 2 species of knowwit in Finland. None of these plant species showed symptoms according to the reports. Here you have the life cycle of the bacteria. When a psyllid feeds on the phloem sap of an LSO-infected plant, the bacteria can be ingested by the psyllids. Then, after that, the bacteria cell must pass through the alimentary canal wall, move through the hemolymph, and finally reach the salivary gland. After this, the bacteria can be transmitted with salivary secretion into a new host plant during psyllid feeding. The bacteria can also be transmitted to the offspring of an infected female psyllid. LSO A and B are transmitted horizontally by Bacteria coquerelli. This psyllid is present in the countries where haplotype A and B have been reported. And as you can see on this map, extract from the HIPPO global database, it is absent from Europe. For Apiaceae haplotypes, haplotype C is transmitted by Triosa apicalis, while haplotype D and E are vectored by Bacteria trigonica. Here, Triosa apicalis occurs in northern Europe in carrot fields. It breeds and feeds on Apiaceae and prefers spurs as an overwintering host. Bacteria trigonica is more present in the southern Europe and the Mediterranean basin, and in the Middle East in Apiaceae crops. More recently, other psyllids have been found to be able to acquire the bacterium by feeding on infected plants. For Bacteria tremblei, it could be shown that it is not able to transmit the bacteria. For Bacteria nigricornis, which is present in Asia and Western Europe, preliminary works being conducted in Spain suggests that infected Bacteria nigricornis is able to transmit LSO. LSO have also been found in other psyllid species. Here, Triosa urticae for LSO-U, Triosa antrixis for LSO European haplotype, and Craspedolepta nebulosa and Craspedolepta subpunctuata for haplotypes Cras. But the ability to transmit the bacterium has not yet been demonstrated. So one of the principal questions, especially in Europe, is what is the risk of transmission of the bacteria between different plant host species. So when the transmission of LSO into carrot plants by Bacteroceras coccarelli was tested, only 1.5% of the carrots exposed to the tomato potato psyllid became infected, and only in laboratory condition, not in field condition. In Spain, colleagues studied the transmission of LSO-E by the psyllid Bacteroceras trigonica and detected 3% transmission rate in potato plants with this psyllid. At the same type of experiment were conducted by Finnish colleagues with Triosa apicalis, and they observed no transmission. Preliminary work concerning Bacteroceras nigricornis suggests that this psyllid, when it is infected by the bacterium, is able to transmit LSO-E to carrot and potato and cause typical symptom of disease. And this psyllid is widely present in Europe, Asia, and North Africa. So further experiments are needed to confirm this, but it's one of the main risks for the moment. Concerning seeds, so potato seed tubers are able to transmit the bacterium, but this mode of transmission seems to be negligible because the new formed plants are very weak and did not survive, so they are considered to be not a source of the bacterium in the field. Although a limited number of experiments have been conducted on Liberibacter transmission, it appears that Candidatus Liberibacter solanacearum is not transmitted through seeds produced by infected plants for Solanaceous plants. And what about Apiaceous seeds? So first experiments conducted between 2014 and 2015 by EVIA and ANSES in the same time with the same seeds batches, but in different agronomical conditions. In France, we applied overwintering in order to reach the next generation and to see the impact on produced seeds. And we obtained different results. The Spanish laboratory concluded that LSO is transmitted by infected carrot seeds, whereas we never detected positive seedlings. Results were published with the impact you probably all know on the international trade on Apiaceous seeds. That many countries made regulatory importation on Apiaceous seeds because of this report. New experiments was conducted in ANSES in equivalent agronomical condition to the ones of the Spanish colleagues, and in addition, different independent laboratories also tried to reproduce the Spanish results. But the results of Bertolini et al. In 2014 supporting seed transmission could never be confirmed in more recent experiments. In ANSES, we never found another positive seedlings, but also the other colleagues in Japan, in Israel, in Finland, and in Italy and another study in Japan. So if you have to take some message at home with this presentation, so what is important to note is that more plant hosts than previously described can be infected by Candidatus Liberibacter solanacearum, and probably if we continue to search, we will probably find more. It is probably the same for the range of vector, and there is still a necessity to evaluate the risk of transmission between different families of plants. The main risk concerning LSO is the introduction of new vector in a country. And finally, if the transmission of LSO by carrot seed is possible, it is probably rare and difficult to reproduce. So I want to thank you all for your attention, and you can find more information on the PO datasheet and also on the EFSA pest survey card. And do not hesitate to contact me if you need.
Thank you very much, Marianne. That was very interesting. I would like to invite now John Clarkson from Warwick Crop Center, School of Sciences, University of Warwick. John is also a lecturer at the Warwick University for plant pathology, and he studied in Leeds and Nottingham. John will present on Fusarium wilt in lettuce, challenges for diagnostics, dynamics, and control. John, please go ahead. Thank you.
Thank you very much, and hopefully everybody can see me okay and hear me as well. Okay, so I've already jumped ahead. So yes, the title of my talk this afternoon is Fusarium wilt in lettuce, challenges for diagnostics, dynamics and control. And thank you very much to the organisers for inviting me to speak to you this afternoon. Okay, so I'm going to start with some background on Fusarium oxysporum. So it's a globally distributed soil-borne fungal plant pathogen, and it causes vascular wilts and root rots on a very wide range of different crop and plant host species. And some of these you can see on the right-hand side. These are some plants that we've worked with here. So one important thing to remember about Fusarium oxysporum is that it's a species complex. So this means it has many different special forms, and each of these are known as forma specialis, and each of these is uniquely adapted to a particular host. So each plant, if you like, has its own individual forma specialis, and as I said, a wide range of plants are infected, including very important horticultural crops such as onion, leek, tomato and lettuce, which I'm going to talk about today. Now, to add to the story, add to the complexity, within each forma specialis, you can get races that emerge that break plant resistance that has been deployed for that particular plant. And one of the challenges in Fusarium oxysporum is that it produces long-lived chlamydospores that can survive for quite a long time in soil. So once you've got a problem, it's very hard to get rid of it. And also within the Fusarium oxysporum complex, you also have non-pathogenic forms. So this adds added complexity, for instance, to developing diagnostic tests. So this afternoon, I'm going to focus on Fusarium oxysporum forma specialis lactucae. I'm going to call this FOL from now on for short. And this is the forma specialis that affects lettuce. And it's present in many lettuce growing areas around the world. And race 1, which I'm going to call FOL1, is the most widespread. And it primarily causes disease in field-grown lettuce and is particularly prevalent in warmer locations such as the United States and Southern Europe, and you can see some pictures of symptoms on the right-hand side showing some wilting lettuce in the field. There are another two races, races 2 and 3, which are largely confined to Asia, which are less significant. But what I'm going to focus on this afternoon is the emergence of race 4, which I'm going to call foal 4. This has emerged very recently and first identified in Northern Europe, and it's causing very significant losses in protected lettuce. So in Northern Europe, and particularly the UK, we haven't been affected by Fusarium oxysporum in lettuce before, but this race 4 is causing significant problems. And like many other pathosystems, the races within foal are defined by the response of a set of lettuce lines to these particular isolates. So in terms of the UK, when did we first begin to observe foal? Well, it was first actually identified in the Netherlands back in 2013, and it wasn't until 2016 that we first saw symptoms. And this was in the Republic of Ireland and also in Lancashire. The growers at this particular time didn't actually know what it was, so it didn't get properly identified. The following year, we had more outbreaks, and this is where we confirmed in the lab here that foal 4 was responsible for these particular symptoms, which you can see on the right-hand side. And since then, we've seen spread to quite a few lettuce growing sites in the UK. Now, the particular varieties that are affected are the little gem and butterhead types. These are the types that are grown predominantly under protection. But I should stress at this point that we haven't found any foal 4 on outdoor lettuce in the UK so far. So it's only at the moment confined to protected lettuce grown in greenhouses or polytunnels. So we had a small project which was funded via the AHDB, which is the grower levy body in the UK. And the growers set us a number of challenges to find out something about this particular race. But so I guess that there had been limited research in this area. So firstly, how do we properly identify and potentially quantify foal 4? So they were interested in, potentially developing a soil test for the pathogen. They were also concerned about what the conditions were for disease development. And we had reports from the growers that they were seeing Fusarium symptoms, not only in the summer, but also in the winter months, so in November and December, when temperatures were a lot cooler. So that was a bit surprising. So there was this thought that potentially race 4 could develop under colder conditions than race 1. They also asked the question of whether non-host cropping could reduce the amount of inoculum of the pathogen. And finally, obviously, the growers here, like in many parts of Europe, are interested in developing resistance to race 4 because the types that are grown indoors are very, very susceptible. So we tried to address some of these questions through our research. So firstly, on the identification. So I think at least the symptoms of Fusarium wilt can be very clear on lettuce. As you can see on the right-hand side here, you get this characteristic wilting and plant death. But I think what's the most diagnostic kind of symptom is if you split open the lettuce longitudinally, as you can see in the photograph on the far right, you get this characteristic vascular browning. So this is a good indication that your plant is suffering from Fusarium. In the lab here, we can confirm by isolation of the pathogen from these type of disease samples, and we have also developed specific PCR and qPCR tests for race 4. One interest of the growers in developing these diagnostics is, can we relate, for instance, the amount of pathogen DNA in soil to the inoculum level and subsequent disease development? I should also say that we developed a rapid LAMP assay. This is, if you like, a very quick version of a PCR, which you can do on a portable instrument, as you can see in the photograph. And you can see the advantage of this particular technique is that you can take some tissue samples from your symptomatic plants. You can make a crude DNA extraction in a buffer, put it into the machine, and you get an answer in about 10 minutes. So these type of machines are actually portable, so you could in principle take these to different grower sites. So the question of inoculum then, in order to try and understand how much inoculum of FOL4 you needed to cause significant disease, we artificially inoculated different soils, which you can see in the graphs here, labelled soil 1 to soil 3, and also in potting compost, which we know is very conducive to Fusarium infection. And as you can see in the graphs, we can get very good dose response curves in relation to the amount of inoculum that we introduce into the soil. The take-home message from this really is that you need around 1,000 spores or 1,000 colony-forming units per gram to cause significant amounts of disease. We could also relate the number of colony-forming units of inoculum that we introduced to a vascular browning score. So at the end of the experiment, we split open the plants and observe the degree of browning inside. And you see we have a good relationship here, as shown in the graph, in each of the soils and in compost. And finally, in this experiment, we also extracted some DNA from the soil at the start of the experiment. So this is from the known amounts of pathogen that we've added. And you can see here that we get a very good relationship between the amount of inoculum that we introduce into the soil, which is shown on the x-axis in this far right graph. We can directly relate that to the amount of DNA which we detect by our qPCR. And in turn, that is also related to the amount of vascular browning. So we do have a way forward, I think, to potentially detect the pathogen in soil and also predict, reasonably accurately what kind of disease risk is posed. So the next question from the growers was, what is the effect of temperature on the development of race 4? So as I said earlier, there was this thought that it could develop at lower temperatures compared to race 1. Now, when we grew race 1 and race 4 isolates on agar at different temperatures, we actually saw no difference, interestingly, in their growth rates. However, when we artificially inoculated lettuce and put them at different temperatures, we did find evidence that FOL4 could cause much more rapid wilt disease at lower temperatures, and that's shown by the graph on the right-hand side. So again, this shows you the vascular browning score at the end of the experiment. And you can see that race 4 is very happy to produce high amounts of disease at 12 degrees C, as well as 16 and 20 degrees C. And it's not till 24 degrees C that we see some infection from race 1. So this is only a preliminary experiment, but it does suggest that perhaps race 1 and race 4 have different temperature requirements, and indeed race 4 can cause infection at lower temperatures. So as I said, the growers were interested in non-host colonization by race 4, because what they wanted to potentially do was introduce a break crop. They were very used to growing continuous lettuce, but they did consider whether they could introduce some other leafy vegetables, for instance, as a break crop, and whether that would reduce the amount of inoculum of FOL4. So we grew a number of different crops in infested compost, and then we measured whether they could be colonized by FOL4 using our qPCR. On the right-hand side here, you see the graph, and you can see that all these different crops you can see on the x-axis here, they did actually sustain some root colonization by FOL4. On the left of the graph, you see that we also grew in this infested soil. We grew some susceptible lettuce, which is the cultivars Amica and Tamira. And hopefully you can see my mouse here. And you can see not surprisingly, we have some very high levels of the pathogen on the roots. However, we also had a couple of resistant lines here, which we selected, and even those are also colonized. So the take-home message here is that even if you're growing a potentially resistant lettuce or another crop, that inoculum of Fol4 can still be sustained in that soil. So it doesn't necessarily decrease the inoculum. We've also seen at some grower sites where some of them have been testing some resistant cultivars that have been on early release, as it were. We've also seen evidence that they are colonized even in the xylem system, as you can see in the photograph there. Although the above ground symptoms weren't too severe, it does look like the pathogen can potentially enter these potentially partially resistant lettuce varieties. So that brings me on to work we've been doing on resistance to Fol in lettuce. Now at Warwick, we're fortunate enough to host the UK Vegetable Gene Bank, and we have thousands of accessions of different crops that include lettuce, onion, brassica and carrot. So we selected 96 lines of lettuce from a lettuce diversity set that we put together that represents the majority of diversity within this crop. And to cut a long story short, we identified quite high level resistance to race 4. And we are developing this in collaboration with Enza Zarden, partly through a PhD, again funded by AHDB, with the PhD student Andrew Legg. So just to give you a flavour of the results from these sort of screening experiments. So this shows you maybe half the diversity set, and along the bottom here you have the different diversity set accession lines. And the y-axis again shows the vascular browning score. So this is the degree of infection by both race 4, which is shown in the blue bars, and race 1, which is shown in the orange bars. And you can see some different lettuce types here within the lettuce diversity set. So you'll see within the butterhead types, and these are the types that are grown predominantly indoors, we see that actually there aren't many accessions there which show much resistance to either race 1 or race 4, although we do have one down here, number 14, which is resistant to race 4 but not race 1. But within the other race types, we see some here where you see some blanks here, so no bars. We have some lines, particularly in the Batavian types as well, which are resistant to both race 1 and race 4. And we have some which are resistant to race 1 or race 4. So just to show you some photographs of these, and it looks like some of these lines have very good level of resistance. So on the left-hand panel here of 4 photographs, we have a line which is resistant to both race 1 and race 4, and the top 2 photographs here are on uninoculated plants. And the 2 plants at the bottom are either inoculated with race 1 on the left or race 4 on the right. And you can see they look pretty much identical to the uninoculated. So there's a good level of resistance. And on the right-hand side, in a similar fashion, we have an accession which is resistant to race 4 but not race 1. So you can see we get very severe symptoms here. This is a line that's susceptible to race 1 and resistant to race 4. So we think we've got a way ahead there in terms of sources of resistance to develop for both race 1 and race 4. Now, through the PhD project, we're doing some other things. So what we'd like to do is to examine gene expression in the pathogen to understand those genes that are expressed in early infection. And we can also potentially look at gene expression at the same time in lettuce plants through an RNA sequencing type experiment. So to do this, we've had to develop, if you like, a bit of a clean system in order to effectively extract DNA. And we do actually see the phenotypes in what is quite a harsh artificial inoculation system. So on the left-hand side, we have a very susceptible line, and you can see the plants are dead. You can see extensive browning of the roots, including the taproot. Here's one of our resistant lines. We get a bit of browning here. But what's interesting is we don't see any significant browning in the main root in lettuce. And this is an uninoculated control plant on the right-hand side. So hopefully what we're going to do is identify some of the key pathogenicity genes, if you like, that are involved in infection, and potentially we might be able to identify some genes that are identified in the resistance response in lettuce. Now, just to wrap up, one concern there has been amongst breeders around Europe is there potential genetic variation in race 4 which could affect potential resistance response. And I think this has come from observations that if you use different isolates of race 4 for your screening, you might end up with a different result. So again, through the PhD project, we've begun to examine some of the molecular genetics of Fusarium in lettuce. So this phylogenetic tree we're looking at the moment, this is based on a housekeeping gene called TEF. It's the translation elongation factor. And the take-home message from this is that when we sequence isolates for TEF, we find that all race 1 and all race 4 isolates have an identical sequence. However, we also observe that the race 3 isolates, race 2 isolates occupy different parts of the tree. Now, this suggests that race 1 and race 4 may have a common origin, and it could be that race 4 has evolved from race 1. I've also picked out at the top, here is a clade that actually contains non-pathogenic Fusarium oxysporum isolates, which we've actually isolated from the vascular system of lettuce. But the take-home message from this particular tree is that TEF is not a reliable way, firstly, of distinguishing different forma speciales. You'll see that they're all mixed up here. And obviously, we cannot use it to distinguish race 1 and race 4 either. What we have done, however, is gone on to sequence a gene called 6H, which we know is present in race 4. So these 6 genes, so-called secreted in xylem genes, are well known to be potentially associated with pathogenicity in Fusarium oxysporum. And what's interesting when we do this is that we find there is variation in this potential pathogenicity gene, even within race 4. And I've picked out 2 groups here, a number of isolates in this green box and a number in the red box as well. So that does suggest there is variation in race 4. And because there's variation in the potential pathogenicity gene that's involved in infection, then perhaps, you know, this could affect the kind of results you get when you're screening different lettuce cultivars. You'll also notice that other Fusarium oxysporum form speciales, also some others contain 6-8, and they occupy different parts of the tree. Okay, so just to summarize, just hopefully demonstrated that diagnostics are particularly important to identify FOL4. It's very useful to know what race you have in a particular country that's causing problems. And we hope that also this may be used in the future to predict disease risk. We have preliminary evidence to suggest that race 4 can cause disease in lettuce at lower temperatures than race 1. We also have evidence that potential non-host crops and even resistant lettuce varieties may allow persistence of race 4. We've identified sources of resistance to both FOL1 and FOL4, and hopefully those will be useful to develop better resistant cultivars. But the potential impact of the genetic diversity within FOL4 still needs to be assessed. And Valerie, who's going to speak to in a moment, has organised a ring test whereby a number of breeders around Europe are testing different FOL isolates and looking at response of different lettuce accessions. So finally, I'd just like to thank my group and the people involved in the work. Hopefully they're all still smiling at the moment. So Nicole, Sasha, Andrew and Alison. And I thank you very much for your attention this afternoon. And now seamlessly, I'm hopefully going to hand over to Valerie, who's now going to talk about the potential for seed as a transmission pathway for Fusarium oxysporum forma speciales lactucae. So I will hand over to Valerie now, hopefully, and I'll change the slide, Valerie, to your next one.
Thank you, John. Yes, it's a question. Is seed a pathway? In all the different subjects we talked today, we spoke about seed transmission. So we have the same question for Fusarium on lettuce. Is it a pathway? Well, the answer is still not proven, because we know that fall is seed-borne. There have been several papers in the literature showing that we can find it on seeds. But is it really seed transmitted? The seed transmission has been shown, but only in artificial transmission experiments where the seeds were inoculated and then there was a sowing. So yes, in this case, in artificial conditions, yes. But what in natural conditions? On the ISF regulated pesticides, it's not indicated because it's not yet known. And there is really no knowledge about seed transmission in natural conditions. So that's why in France, there has been a project initiated. So the name of the project is Actifol, which is coordinated by CTIFL and in collaboration with my lab in GVS and seed companies to be able to conclude if seed is a pathway or not. So it's under progress, and the experiments are done by inoculation of mother plants with variety which are susceptible, intermediate resistant, and resistant. It's for fall 1 in this case, with harvest of seeds, and then we will do some detection on media. And if we find in natural condition some Fusarium on the seeds, we will do some grow outs, like here in this picture, but here it's a resistance test. Just to see if when the seeds are sown, there is a transmission or not, but more in a natural way of the seeds. So that's still under progress, and I hope that we will have the answer in 1 or 2 years. Thank you for your attention.
Thank you, Valerie, and thank you, John, for your nice presentation. I will now introduce the last speaker, that is Stefania Loretti from CREA, which is the Council of Agricultural Research and Economics in Italy. And Stefania is talking about Xylella fastidiosa, general traits of a recently introduced ancient threat. Stephanie, thank you for joining us today, and please go ahead.
Thank you. Can you see the presentation?
We can well see your presentation, Stefania.
Okay, thank you. First of all, I would like to thank the organizers to give me the opportunity to have this talk. And the presentation will provide an overview of the main characteristics of Xylella fastidiosa. And part of the outcomes here presented are the results of the activity of the XF Actors project. Tracing the origin of this disease, the first report of a disease caused by Xylella fastidiosa dates back to the end of the 1800s on grapevines. And the first report was from Newton Pierce, the scientist who gave the name to this disease, currently known as Pierce disease of grapevine. But after about 80 years, there was the first isolation when was recognized a bacterium as causal agent of the disease and not a virus as previously suspected. And about 10 years later, there was the formal description and naming of Xylella fastidiosa to the causal agent of the disease, where the name Xylella is associated with the xylematic location in the host, and fastidiosa with the in vitro growth difficulties of this bacterium. In the meantime, several other urban and forest plant trees were found to be infected in the eastern USA. And at the end of the 19th was described the citrus variegated chlorosis in Brazil. And it is worth mentioning that the first genome sequencing of a plant pathogenic bacteria was a strain of Xylella fastidiosa affecting citrus. It is a gamma protobacteria, which it belongs to a gamma protobacteria, is a gram-negative bacteria. This class is reported in this group in orange, where are reported the main genera of bacteria affecting plants. And the most related, from a taxonomic point of view, to Xylella is the Xanthomonas. Is a xylem-limited bacterium which move in a cropetal direction, but it can move also against the xylematic flow reaching the roots by twitching motility. It forms biofilm that may plug the vessels. It is transmitted by sap-sucking insect vectors among plants, and dissemination at large scale occurs by plants for planting. Xylella switches its phenotype from a planktonic state in which it colonizes the plant host and moves within the plant host to a biofilm state in which the cells adhere to the plant cell walls and aggregate each other, resting. And at this stage, at the biofilm stage, it is acquired by the insect that promote new infection and the dissemination of the bacterium. About the subspecies, from a scientific point of view, are known 6 subspecies where the tasks were no more recognized. The International Society of Plant Pathology for the taxonomy of plant pathogenic bacteria recognized only the first 2, but recently, on the basis of comparative genomic analysis, has been recognized as the first 3, where the subspecies Sandy and Morus are included in the first one, the subspecies fastidiosa. If we look at the host, the main host, the subspecies fastidiosa, fat, grape, alfalfa, almond, but several other plant species. The multiplex has the wider host range. It affects up to about 150 plant species. The pauca have a restricted host range, and the main diseases are on citrus, coffee, and olives. And the sandy and the morus have a restricted host range on oleander, mulberry, and blueberry, respectively. Within each subspecies, Xylella strains can be further characterized in sequence type on the basis of the sequencing of 7 housekeeping genes. And currently are known 87 sequence types in total. Why it is important to know the subspecies, but also the sequence type? Because whenever a new recovery of Xylella fastidiosa occurs in a pest-free area or on a new plant host, the subspecies, but in particular the sequence type characterization, give us information on the presence of possible recombination among strains, which drive the evolution and adaptation of Xylella to new plant host, and give also some information on the possible origin of the strains. The current EFSA report mention about 600 of different plant species as host of Xylella fastidiosa, and the main symptoms is the hydric stress that can be in which we can be observed a leaf scorch that evolve in wilting of twigs and branches, and then in debulk and death of the plants. Another kind of symptoms caused by Xylella is the stunting and dwarfing, which that can determine plant growth arrest and can be observed on alfalfa, on citrus, and on peach, as we will see in the next slides. The symptoms of hydric stress is due to the occlusion of vessels of the plant host, and these are determined by several aspects: bacterial cell aggregation and isopolysaccharide matrix that determine the production of biofilm that can be shown in this slide, in this imaging. And then the overproduction of defense compounds, pectins, and also the formation of tills that are produced by the host plants in response to infection. And then the formation of high bulbils following insect bites and water stress phenomena. And then the production of anamorphous gels that coat the xylem vessel, and it is of plant origin. All these factors contributes to the occlusion of vessels. Looking at the main disease, we have to mention the Pierce disease of grape, in which can be observed leaf scorching that evolve, causing irregular spots on vegetation. It can be also observed the presence of green areas on lignified branches, then grape desiccation, and that of the plant. It has been estimated that the losses for direct damage caused by the bacterium and the prevention cost implemented for the control is amounted to about $1 million per year. Plants affected by citrus variegated chlorosis showed a reduced vigor for shortening of internodes and a desiccation of distal portion. Leaves showed internalveolar chlorosis, and fruits are reduced in size, shows an early ripening, high sugar content, and a leathery consistency. Also, in funny pitch disease can be showed a reduced development due to a shortening of internodes and an increasing of lateral branching. It can be also observed darkening of leaves, early flowering and ripening, and a reduced of fruit size. Infected coffee plants have been intercepted several times in Europe, and it seems that Xylella fastidiosa has been introduced in Apulia region through asymptomatic infected seedlings marketed for ornamental purpose. Symptoms are leaf scorching, internode shortening, but also symptoms of crespera, in which can be observed curling of leaf margins, chlorosis, and deformation of leaves. However, these symptoms are not always associated with the infected plants, which can also remain asymptomatic. And finally, leaf scorching that can be observed in a very quite high number of plant species. Here are reported symptoms on elm, maple, ginkgo, almond, and plane tree, but can be also observed on several plants of the Mediterranean scrubs, and has rosemary, cistus, brome, and also on ornamental plants as polygala, mimosa, lavender. Despite the symptoms highlighted in the image just shown, it is important to underline that infected plants can remain asymptomatic despite being infected. And on the basis of experiment of artificial infection performed at the INRA in France and from the CNR of Bari in Italy, it has been observed that the length of the asymptomatic latent phase is variable according to the bacterial strain and the host, and range from 2-3 months in the combination grape subspecies fastidiosa up to 14 months in the combination olive subspecies pauca. Xylella is a quarantine bacteria, and it is reported among the pests known to occur in the Union territory in the current regulation and in the A2 list of EPO. Here is represented the EPO map of the distribution of the pathogen worldwide, and it is possible to see how it is quite common in the Americas, but also in Europe, and it is also recovered in Israel, Iran, and in Taiwan. Looking at the distribution of the subspecies, we can see that the main subspecies, these 4 subspecies, has been recovered in the Americas and then in Europe, whereas only the fastidiosa is present in Iran, Israel, and Taiwan, where it is also present the Xylella taiwanensis, another species of Xylella. The introduction and spread of the different strains took place through mechanism of recombination and translocation between plant hosts. It has been observed that the subspecies fastidiosa and the subspecies sandy both originated from Central America and were introduced in the USA at the end of the 18th, the fastidiosa, and at the end of the 19th, the sandy, respectively. And Xylella fastidiosa subspecies multiplex was introduced from the USA to Brazil, where it would recombine with the native strains of the pauca, generating the current strains responsible of the citrus variegated chlorosis and the coffee leaf scorch. And it is worth mentioning that another divergent strains from the pauca responsible of disease on coffee and citrus is that one able to cause disease in Italy on olive and causing the so-called olive quite decline syndrome. Looking at the chronology of the reports outside the Americas, we can see that the first real introduction of Xylella in the European territory was that one of 2013 in Italy, in Apulia region, and then in France, in Corsica and in mainland, then in Spain and Balearic Island, and in 2018 in Portugal, and again in Italy, in another region, in Tuscany. In all these cases, there was outdoor inspections of Xylella on groves, cross-inter landscape and orchards. Whereas in 2018, several interceptions in indoors material were recovered in Belgium, Holland and Germany. And finally, the subspecies fastidiosa was recovered from 2013 to 2018 in Taiwan, Iran and Israel on grape and or almond. Another important aspect to take into account in order to understand the possible relationship between the strains and the possible recombination among strains and possibly the origin of the strains introduced in a new area is the distribution of the sequence types. If we look at Italy, we can find a quite homogeneous situation in which in Apulia region there is only the pauca subspecies and one sequence type, the 53. If we move to Tuscany, we found only the multiplex with a new sequence type, the 87, that was characterized within the X Factor project. So only two subspecies with each one only one sequence type. Moving to France, we can find a more heterogeneous situation in which we were recovered three subspecies: sandy, multiplex, pauca. The multiplex is the most widespread with the sequence type 6 and 7 that were well known in the USA and were recovered here on Polygala and elm. And also is present the pauca and the sequence type 53 that is the same of Apulia region, but it caused disease not on olive, but on Polygala and on almond. And it is quite divergent from this one. Another characteristics is the recovery of a new sequence type, the 71, that it is the origin of a recombination among the subspecies sandy and multiplex. Moving to Spain, we found a very heterogeneous situation because are present three subspecies, pauca, multiplex and fastidiosa. But it was found that, for example, in the same plants, in Polygala plants, are recovered up to 4 different sequence types in coexistence. The most widespread subspecies is the multiplex, where the sequence type 6 is slightly divergent from that one in France. Moreover, in Spain, were individuated disease on olive, but it is caused not from the same sequence type recovered in Apulia region, is a different one. And it is also it is in olive plants where was also recovered a strain of the subspecies multiplex. On almond, there is a very severe disease that is called golden death to the yellowish of the vegetation. And it is caused by the presence of the multiplex with the sequence type 81 and the fastidiosa, which in the Balearic Island cause disease also on grape one. So a very complicated situation. Moving to Portugal, we found the multiplex and the sequence type 7 only. If we look at the host plants, we can see that in all affected areas, there is a large impact of the disease on ornamental plants and Mediterranean scrub in all this area in which Xylella was recovered. Finally, and I conclude, some information about the evolutionary correlation between the European and the American subspecies. It was studied, and the study showed that Xylella fastidiosa subspecies multiplex was original of the southeastern USA, and strains from California, Europe, and Brazil are a consequence of different introduction events. In particular, strains of the subspecies multiplex in Europe seems to be introduced from California, and recently has been found 5 different introduction events in Europe. That one of Italy in Tuscany region, in Corsica, in Spain, in Balearic Island, and in Alicante. In all these cases, this regards the subspecies multiplex. Moreover, it was shown that all these strains belong to a relatively recent group, so are of recent introduction, between was found a limited recombination among these isolates. And the same was for the Xylella fastidiosa subspecies pauca recovered in Apulia that seems to be linked to a single introduction event from Costa Rica. And also in this case, the introduction seems to be relatively recent. Thank you very much for your attention. I conclude my presentation.
Thank you very much, Stefania.
Okay.
We open the area now for everybody again. I saw that some of you were looking at their questions which were directly related to them. Could we get all panelists on the screen here? So that there were a few questions to John as well. I think you tried to answer them in the background, but could you read your question to us, John, which was passed on to you?
Yes, I found one question. I haven't seen any others. And the question was, is there a recommendation to reduce the amount of Fusarium inoculum in the soil? I think that's challenging to do, but I do know that some protected lettuce growers, not necessarily in the UK, but certainly in the Netherlands, have used soil steaming to sterilise their soils. That is effective, but I think they see the disease beginning to return in the 3rd successive crop post-steaming. The other option is a soil fumigant called Dazomet, but certainly its use is severely restricted in the UK and probably elsewhere as well. So I'm not sure of the future of Dazomet in terms of approvals in Europe going forward. And finally, I think for field-grown lettuce, I think in the United States they just employ long rotations, or they try and grow the more susceptible cultivars at times of year that are less conducive to disease, so at cooler times of year.
Thank you, John. I've got just a question which came in for Stefania. And the question is it possible that a plant can be infected with 2 different subspecies of Xylella?
Thank you for the question. I think yes, it's possible, because it is possible also because may occur recombination among different strains, subspecies of Xylella that generate new sequence type. The example was the sequence type 71 in France That was the results of a recombination among sandy and multiplex. So probably the two bacteria have the possibility to recombine, and then it is also possible that can affect the same plant.
Thank you. I've got a question for Rob here. Rob Tanner, how do you consider that the invasive pest is high risk category?
Yep. So if we're taking the prioritization process and to a large extent the evaluation of the pest risk analysis process, it will all be based on evidence and expert opinion. You would score different sections, and so high-risk species, for example, could be those plant species that are spread long distances by wind dispersed seed, coupled with the biology of forming dense monocultures that can outcompete native species, impacting agricultural systems, have the potential to establish in habitats, endangered habitats. Therefore, what I'm trying to say is you take into consideration all of the factors within the pest risk analysis to come to the overall conclusion. But evidence, evidence, evidence. It's based on evidence, and where evidence is slim, it is based on expert opinion from across the scientific sector, from pest risk management experts, from biologists to ecologists, etc.
Thank you very much. I've got a question for Robert. Don't mix up. Are TOBRFV resistant tomato varieties able to infect other susceptible tomato or pepper varieties?
Yes, that's a very good question. And I think that there's no doubt that the varieties with intermediate resistance that are symptomless carriers will be sources, potential sources of inoculum for susceptible tomato varieties. So again, if even though they are showing no symptoms, these intermediate resistant varieties do have the virus, and we believe a fair amount of it. So simply touching those symptomless plants and then touching a susceptible variety nearby will lead to infection and symptoms in the susceptible variety. So that's a big concern with these intermediate resistant varieties that don't show symptoms but still carry the virus. So that is a big concern because they definitely can be sources. Now, the extreme resistance, where there's little replication and movement, would not be the case because the virus is not able to spread in those varieties. But the currently available resistant varieties that are commercially available are these intermediate resistant ones. So hopefully these more extreme resistant varieties are coming in the near future.
Thank you, Robert.
Andreas, I see another question for me. One was, how different are symptoms of GBNV and tomato brown rugose? GBNV is groundnut bud necrosis virus, that's found in India and the subcontinent in Asia. The big difference there, it's a tospole virus, thrips transmitted. You're going to see necrosis in the foliage. Okay, so any tospole virus in tomato tends to cause necrosis in the foliage, bronzing, also ring spots on the fruit. But the foliar symptoms of brown rugose do not involve necrosis. They only cause a mottling, mosaic, a leaf distortion, crumpling, but no necrosis. And then you can, if you suspect you have brown rugose, then you can use the immunostrip test, the lateral flow device, either for a tabamovirus, TMV, or for brown rugose to confirm the diagnosis. But those, the Tospol virus versus Tabamo virus symptoms are pretty different in the foliage.
Thank you very much. I got another question to Rob. What is your comment about the fact that grains are required to be free from amaranthus seeds in the grains if the grains are not used for propagation?
Yep. So in the EPO-PRA, we evaluated the two main pathways. One was grain, animal feed. The second was seed for planting. The grain pathway, there are a lot of reports of association on that pathway. The likelihood of it reaching a suitable habitat, i.e. deposited within a farmyard or in a farming system, is high. And therefore, within the EPO-PRA, we did recommend measures for grain as a pathway due to the high association, the likelihood of entry, and the likelihood of reaching transfer to a suitable habitat. The measures were pest-free area or inspection using ISPM 31, or the devitalization of the product. And so to answer the question, I think the measures especially are reasonable due to the high risk of the pathway.
Thank you. There was one question also to Marianne. I think you got that question. Could you give us the answer to that and read the question out, Marianne?
Yes. The question is the LSO vector can travel in long distance, and what is the method to control the vector? So in USA, to speak about Bacteria coccolelli, they evaluated that adults are very good flyers, and they can and mainly because of the winds and hot temperatures, and they evaluated movement of this psyllid can be in a range of 100 kilometers. But the other stages, the life stages of the psyllid are sedentary and do not actively move. But they can be dispersed in long distance by human activity and transport of solanaceous plants. For example, you can find nymphs or eggs on solanaceous fruits, mainly if you have green part on these fruits. And to answer for the control strategies, again, vectors, it exists chemical controls. So when it is necessary, they apply mainly chemical controls. And I know that different projects now investigate the possibility to use natural predators.
Thank you very much, Marianne. And I've got a final question to Robert. How should I take samples from transplant for TOBRFV?
You know, that's a tough question. You know, you would have to establish a random kind of pattern of selection of plants, because as I mentioned, at least as far as I know, it's very rare that the transplants show diagnostic symptoms. So you would have to set up, do some random sampling, and then I would just grind leaves from those plants and rub inoculate those tobacco indicators. But again, I would say, you know, you could be also walking through your transplant house and looking for unusual symptoms, but it's very hard in the transplants. So, you know, it would have to be some method like that, but, you know, very difficult. And again, focus on the seed, starting with clean seed, and you're going to be quite safe that you're going to have clean transplants. That's the best way to go about it. I saw a couple other questions too, Andreas. One question is very important. What's the best way to detect to do seed health testing for tomato brown rugose? And the question is ELISA or the bioassay. Well, if you're going to pick those two tests, ELISA, of course, is enzyme-linked immunosorbent assay. That's not a good method right now anyway, because there's not a good antibody for tomato brown rugose. So if you don't have access to the RT-qPCR, which is the gold standard test, then you definitely want to use the bioindicator, the bioassay with those tobaccos like Nicotiana glutinosa or Nicotiana Havana. And then just grind up seeds and inoculate those tobacco indicators, and you see the local lesions. So if those are the only two options you have, definitely go with the bioindicator. And then the last question was, what's the best time to treat seeds with triple sodium, tomato seeds with triple sodium phosphate? Pretty much any time. Once you've you know, recovered the seeds from the fruits, washed them, dried them, you can then treat with triple sodium phosphate anytime. And it inactivates the virus because of the high pH. It disrupts the protein shell, and then the RNA inside degrades. So it will maintain its protection for as long as you store the seeds. Those were very good questions.
Thank you very much. There are a few other questions coming in here, but due to the time, I would hand over to Valerie for some closing remarks. Not forgetting to invite you to our next session next Wednesday, same time, on new detection methods and alternative control measures. Thank you from my side, and thank you to you, John, Marianne, Rob, Robert, and Stefania. Handing over to Valerie for some closing remarks. Thank you very much.
Pleasure.
Thank you, Andreas. Well, I want to thank our very good lecturers today. It was very interesting to see how these research and results and how we are dealing with new threats and emerging pests. And I really thank you. And thank you also for participants. I know that some of you are connecting from everywhere in the world it's very late for you or very early. So thank you everybody for listening. And next Wednesday, we will speak about alternative control measures and how to deal with these pests. So thank you very much.
Thank you very much. Okay. Bye, everybody.
Thank you and goodbye. Have a good day.
Bye-bye.
Bye-bye.
Bye.
Bye.
Bye.